turbo engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
questions,
Research.
technology.
Unbelievable.
Without further ado,
This is The Naked Scientists.
Hello and welcome to The Naked Scientists, the show where we bring you the latest breakthroughs in science, technology and medicine, with me, James Titko.
And I'm Chris Smith. Coming up this week, a UK toddler with a rare genetic disease is saved by gene therapy. We explain how it works. Cockatoos wow scientists by taking a toolkit along to solve a problem, and a 55 million year old penguin fossil. And it's a whopper that weighed in at 150 kilos.
The Naked Scientists podcast is powered by ukfast.co.uk.
This week, scientists announced that a toddler with a rare, fatal genetic condition has been treated successfully with a new gene therapy approach. 19-year-old Teddy has MLD, that's metachromatic leukodystrophy. It's an inherited disease linked to a defective gene that normally codes for an essential enzyme that's called aryl sulfatase A. Without this working, material builds up inside certain cell types and it causes them to die. The developing brain is particularly vulnerable and affected individuals become progressively more disabled, and they usually don't make it, sadly, past the age of 5.
In Teddy's case, the Royal Manchester Children's Hospital's Rob Wynne and his colleagues have, they think, hopefully effected a cure. They've taken stem cells from her bone marrow and altered them in the lab to add back in a healthy copy of the gene, as well as some control information to supercharge its activity, leading to high-level production of the missing enzyme. Those modified stem cells will return to Teddy, where they migrate to the bone marrow and produce blood and immune cells. Some of these then migrate into the nervous system, where they pump out the enzyme for other cells to use.
In illnesses like MLD, where there is a deficient enzyme, we can use bone marrow transplantation to help the condition. And what happens there, the normal healthy bone marrow makes normal healthy blood, makes the enzyme and delivers that enzyme around all tissues of the body, including in the brain. We know when we do a bone marrow transplant in MLD, actually it doesn't correct the condition, and we've understood in recent years that's because normal healthy blood does not give enough enzyme in this condition. So we take blood stem cells from the patient, and in the laboratory we insert the missing copy of the gene. Now we do that using a virus, but we have modified that virus so when it infects the blood stem cell, instead of delivering virus DNA, they deliver the enzyme gene. But we've also made one other key difference. Genes in our cells are controlled in a very tight way, and actually the MLD gene is controlled in a very tight way in blood stem cells, and this is why a normal bone marrow transplant does not give enough enzyme, because the gene is turned off. The blood doesn't need to make a lot of enzyme. So when we use gene-modified cells, not only have we put in a healthy copy of the gene, but we have changed the promoter next to that gene that tells the gene to be turned on. And so the gene-modified stem cells make vastly more enzyme. And because more enzyme is good for the disease, then we actually see that this makes the illness better, whereas a normal bone marrow transplant doesn't.
These are bone marrow stem cells, so they're going to be making elements of the blood and they're going to be outside the brain. There's a barrier in place that keeps the brain tissue and the blood tissue compartment separate. It's called the blood-brain barrier. So how does making the bloodstream awash with this missing enzyme get the brain treated?
This is an excellent question. It actually goes to the heart of why bone marrow transplant and cell therapy corrects neurologic enzyme deficiencies in a way that drug therapy does not. Cells can cross the blood-brain barrier and engraft, and white cells become engrafted in the brain where they're known as microglia. So if we do a gene-modified blood stem cell transplant, then we will get gene-modified white cells. They will cross the blood-brain barrier and then become engrafted in the brain, and so gene-modified, brain-engrafted microglia make the enzyme that the patient needs, and forever.
And when they're sitting in the brain, they're presumably secreting the missing enzyme into the melia there, and what the dependent cells that would normally be missing it can pick it up and use it.
This is exactly the principle of transplantation in this type of disease, and it's known as cross-correction. The engrafted donor cells make and secrete enzyme. That enzyme is taken up by neighboring cells that remain genetically enzyme deficient, but they're able to take up this secreted enzyme and metabolize within their cytoplasm accumulated substrate.
Is the prognosis then for this young lady a very good one, in the sense that as long as she stably continues to have these cells making this stuff in her brain, then she should remain well?
We think that this girl can look forward to a normal life expectancy and a normal quality. So clearly, using gene-modified stem cells in MLD is new. However, we have a wealth of experience of using transplants from donors in similar diseases. And what we see in those illnesses is that the effect on the condition is lifelong. So we have every reason to be optimistic that the gene-modified stem cells will also similarly dramatically change this young girl's life.
Are there any risks here? In the past, when people have used viruses to change stem cells in this way, they've been concerned that when you're adding bits of DNA to a cell's own DNA, you can make cancers. And also, if they are churning out huge amounts of this material that they wouldn't normally churn out into the bloodstream, are there any health risks from that?
Certainly, all of our medical decisions are based on balance of risk. This is a disease associated with premature death, and children are dying in early life, and before dying, they lose their skills that they had acquired. So, risk balance is very much in favor of this treatment. Viruses insert their DNA into our DNA, and this is why there is some cancer risk if they insert their DNA next to what we call an oncogene that's associated with controlling cell proliferation. They can cause cancer, but the virus vector, as we call it here, is so modified that it inserts randomly into the genome. And if it inserts randomly, then it's much less likely to cause cancer. In terms of the enzyme overexpression, that's a very important point. The very basis of this treatment is that the gene-modified cells make much more enzyme in the blood. So in our preclinical work, we have been able to show that that enzyme expression, that enzyme overexpression, as we call it, does not affect blood stem cell function. And so the cells are able to engraft, and the cells are able to make blood that is otherwise normal.
Amazing story, isn't it? And incredible how far that technology has come in just the last about 2 decades. That was Rob Wynn there.
Very clever stuff. Now to the animal kingdom, though, and an equivalently clever cockatoo species. Goffin's cockatoos live in parts of Indonesia, and they've been found to be capable of one of the most complex pieces of animal thinking ever encountered. They use tools to complete tasks, but not just one type of tool, several. And they can even plan ahead and take just the right number of different tools to get the job done. If only my builder could take a leaf out of their book. Will Tingle takes up the story.
Tool use, or using an external object to change something about another object, is very commonplace in our lives. Chopping food with a knife, that's tool use. Driving a car is tool use, albeit a very complex tool. And tool use features in the animal kingdom too, but it's a rare piece of cognitive behaviour. It's only been observed in 32 species so far. And even still, there's a step above tool use, which is tool sets. That is to say, using 2 or more tools with different functions together to achieve a single outcome. And that's even rarer still, as the University of Vienna's Antonio Osuna Mascaro explains.
There are only 2 species out there in the wild able to use tool sets, or that are using tool sets out there. One is chimpanzees, and the other is Goffin's cockatoos.
Goffin's cockatoo is a parrot that lives in Indonesia, and they're a big fan of the stone inside a fruit of the sea mango plant. But the fruit's flesh itself is very toxic, and the cockatoo's beak isn't strong enough to open the stone. So what to do? Well, the cockatoos have been employing a form of tool manipulation that might put even the chimpanzees to shame.
What the cockatoos did by surprise was to use 3 different kinds of tools to have access to this stone. The first one was the use of a tool with the function of a knife. They used this to cut in between both halves of the stem of this fruit after removing the flesh of the fruit. Then they use a wedge to separate both halves slightly. And then when they have this opening, they use a tool with the function of a spoon to be able to eat the insides of this stone. This is a very impressive kind of tool use and could be the most complex case of tool use out there in nature.
So using a knife, a wedge, and a spoon to get to their food is impressive enough, but that's only a half of the cognitive conundrum. Do these cockatoos know which tools are needed for each step? And if so, would they only take the necessary amount of tools if one of the steps was removed?
We were having 5 birds being tested, and out of those 5 birds, 4 of them were able to transport eventually, and 3 of them were able to transport in a flexible way, even when this required flying with both tools. And that was amazing. That means that they were picking up and transporting both tools together every time that they were facing a problem that requires the use of both tools. And they were picking up both tools only sometimes when they were facing a problem that doesn't require both tools. So they were able to categorize both tools as a tool set. As we like to say, for them, a tool set is more than the sum of its parts.
Goffin's cockatoos then have the ability to not only understand which tools are necessary, but which ones sometimes aren't. And that is something that's pretty unheard of in the animal kingdom. So what's the reason for these cognitively confident cockatoos?
Goffin's cockatoos are a very particular species. They learn to use tools in the same way as we do, and they learn by combining objects, as toddlers do, by playing with different objects and combining them, and then eventually finding functions and ways of using tools. We have 18 individuals in our lab, and many of them know how to use tools. But it's interesting to see how each single individual use tools in different ways. They have different techniques, different approaches. So each one of them have a different way of using tools and a different approach. And that is telling us that they are using their general cognitive capabilities to use tools. They are flexible enough that they can learn to use tools.
The ability to use their claws and beaks to personally interact and combine tools allows them to form ideas as to which tools would work best and in what order. This means they have a more complex knowledge of tool manipulation than any other animal besides us. Such behavioural complexity allows the Goffin's cockatoos to be outstanding in their field. Well, more likely their tree.
So, being bird-brained is more of a compliment than an insult, it turns out. That was Will Tingle who was speaking to Antonio Mascaro, and that paper has just been published in Current Biology.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound, perfect music for audio and video productions.
You're listening to The Naked Scientists with Chris Smith and with James Titko, and still to come, researchers uncover the remains of the world's largest penguin, twice the weight of the average human. But first, seed carriers that can drill themselves into the ground to boost the success of sowing rates have been engineered by researchers in America. They've taken their inspiration from a grass species called Erodium, which has a corkscrew-shaped seed with a sideways pointing anchor tail at one end. When this lands on the ground, penetrating moisture swells the tissue, triggering the spiral to unfurl against the anchor, driving the seed into the ground. By working out how nature does this, Carnegie Mellon scientist Lin-Ning Yao has developed a way to copy and improve on the process, using small pieces of wood that can then be loaded with a seed cargo and deployed as aerial seeding devices, for instance, for regenerating tracts of woodland or conservation areas. Seeds that plant themselves in this way are much less likely to become mouse and bird food or dried out by the sun.
We got very inspired by seeds called Erodium. It's actually a grass. The seeds, once they are dispersed, basically they can dig holes and bury themselves into the ground. Basically, the seed pod has a moisture responsive body, and this body will unwind itself when the moisture level is rising up. And this unwinding and winding motion, basically it spins and self-drill into the ground. It's a little bit like how our electronic hand drill works.
So it is literally a seed drill. That's what farmers use, isn't it, to plant seeds? That's intriguing. So it naturally just bends and changes shape in response to changes in ambient moisture, and that causes a twisting, and that's what pushes the seed in. So you're saying you want to copy that?
Exactly. Yes, we are trying to find ways to engineering replicas.
First of all, let's start then with how nature does it. How does the dampness or the moisture content cause the natural erodium seed to change its shape in the way that it does, to create that twisting or drilling motion for its seed?
The natural seed can self-drill because of its material, but also the structure. So the seeds are made of cellulose-rich materials. It responds to the moistures, traps water molecules into its own body and swell. That is number one important thing. So the number two is the seeds has a coiled body. So as the water molecules being trapped into the material, the coils loosens itself. As it loosens, it spins, and that spinning creates effective vertical downward forces for the seed pod to bury itself into the soil.
And that's what you were seeking to make a replica or an equivalent of. Did you do it the same way?
On the material side, you need a stiff material. You need it to be biodegradable because we don't want to retrieve the seed carriers. We started to think about wood because it's a very abundant, accessible biomaterials that are cellulose rich and also are known to have this moisture responsive behavior. But we had to compare different kinds of wood. Eventually, we chose white oak. We also have to think about the geometry. So one thing we did a little bit smarter than the natural erodium seed is that we are introducing 2 extra tails. So basically, the natural erodium only has a single tail. So when you drop it on the floor, it tends to land flat. But in order for it to drill, it has to lift its own body up by leveraging some crevices on the ground that limit the type of soil erodium can function in. So in our case, we introduced 2 extra tails. So when you drop our seed, they tend to land on the floor with a specific angle, and that angle helps improve the drilling efficiency.
How did you make these things? When one pictures this, I mean, the erodium seed, it looks like almost like a little mini windmill with the seed at the bottom of a stalk. How did you make that? Because you've effectively copied that you've got the 3 arms coming off of yours. How do you make it?
Basically, it has 2 major steps. First, we need to process wood into veneers and then cook them in a chemical solution to make the wood more compliant. And the second step is we mold it on top of a 3D printed mold that we tailored beforehand. Basically, we have to wind it into the coil manually.
Got it. So you 3D printed the geometry you wanted, that you knew ought to work the right way, and then you mold the wood onto that. It's almost like forming it onto a mold to make it adopt that shape. And then you've got your seed carrier, but made of white oak.
Exactly. Thank you so much for helping me to explain this.
How big is each one?
We make a range. So the smallest could be maybe 1 centimeter in height, and you can make all the way about 10 to 15 centimeter in height. And again, this highly depends on what seeds or other things you want to carry with the carrier. Small vegetable seeds only need a very small carrier, but pine seeds, for example, are much larger. You need larger body to carry.
How did you test it to find out if it would really do what you were hoping it would?
We have done both in lab controlled tests, and eventually we also brought it outdoor. So we've conducted 5 rounds of tests with hundreds of seeds in 2 geographic locations. We watched the weather broadcast, Right before heavy rain would come, we threw them on the floor and then we monitored how effectively they can drill.
And what was the germination rate when you did this compared to if you just threw the seeds on the floor?
Across the 5 batches, we got about a 66% anchoring rate. And among the ones that anchors, about 60% of germination rate.
And what about if you just threw seeds on the floor, the same sorts of seeds? How many of those would successfully germinate?
I really wanted to conduct this test, but we haven't got a chance to do this exact comparison. But we do know if you try to germinate pine seeds, if you do broadcasting from the airplane, the germination, 2 to 10%.
So potentially a considerable boost to germination rates. Lining Yao there, and she just published that work in the journal Nature. The news over the past few weeks has been awash with stories of unidentified objects in US airspace, including one that's dubbed the Chinese spy balloon. Recently, the two countries have had a tenuous relationship at the best of times, and this incident looks to add further strain. But how has the situation unfolded so far? James.
I'm sure you'll all have heard this week of the Chinese surveillance balloon that the US shot down, which they now claim was able to collect communication signals. Equipped with instruments capable of intelligence collection operations, it has sparked a major international row. Even more instances have been happening recently, as 2 other flying objects have been shot down over the US. These 2 have not had their origins confirmed yet, and have been described by US intelligence as benign, much smaller in size and not controllable from the ground, so only time will tell if their motives were nefarious or otherwise. As for the main balloon, details remain scant, but it is believed it took off from a base on Hainan Island along China's southern coast. It was spotted at 60,000 feet in Montana before being shot down by a missile for violating US airspace. The Chinese government insists that the balloon was a civilian airship used for research, but American intelligence has since confirmed from the debris that the balloon was being used for surveillance. However, it's now uncertain as to whether the balloon was meant to enter US airspace in the first place or was blown off course by strong winds. The balloon was tracked since its launch and made unusual turns, first into Canadian airspace and then down into US airspace. The wind could have played a part in its course, despite the US claiming that China had partial control of the balloon's movements. So if a balloon is so easy to spot and difficult to control, why use one in the first place? Well, a balloon's movement pattern is harder to track than satellites, as they move more unpredictably and are often smaller than a typical radar's level of calibration. They can also hover above a certain point for much longer than a satellite. Satellites are also tied to their gravitational orbit around the Earth, which is not helpful if you want to monitor the ongoing progress of something happening on the ground. Balloons sometimes dip under weather systems that could otherwise block a satellite's view of an area, and in this balloon's case, it can be used to listen into devices. The balloon, confirmed to have surveillance equipment attached, had technology capable of locating communication devices and listening in to their conversations. But the conversations of who? Civilians? Governing bodies? That all remains to be seen. The US is taking in all of the downcraft for extensive analysis. There are, however, experts on the subject of surveillance intelligence who remain surprised as to why this individual balloon incident is gathering so much traction. Both countries already have extensively detailed maps of one another, and there isn't much a balloon could tell either side. But balloons have been a part of Chinese surveillance for some time now, and the US now has to revisit its policy on how finely it scans for foreign aerial objects, and how they will deal with them in the future. All that can be said for certain is it has been a diplomatic disaster for both countries.
And we will keep a weather eye on what happens with that story, and we'll keep you posted. Now, the fossilised remains of one of the world's largest and earliest penguins have been uncovered in New Zealand, found in boulders on a beach near Otago back in 2016, and it's taken that long to recover them and expose them and now publish what they've found. These date back over 50 million years, and they've now been carefully exposed so that the stature of the original animal can be appreciated and studied properly, and we can work out what it would have looked like and how big it was. And speaking of which, it would have weighed in a hefty 150 kilos. One of the team who is behind this discovery and what this discovery can tell us about the origins of penguins and their subsequent evolution is Daniel Field, and he's at the Department of Earth Sciences in Cambridge. Welcome to the programme, Daniel. How do you know this is a penguin?
What we're talking about here with this giant penguin are the remains of its humerus, or its upper arm bone. And penguin humeri are very distinctive. Of course, penguins move in a very different way from most other birds. They use their wings to fly underwater. And that specialized mode of locomotion has resulted in the evolution of a very distinctive humerus with an offset shoulder connection. The humerus itself is flattened, unlike a round cross-section like you see in the wings of flying birds. And so there's really no mistaking the humerus of a penguin when you find one.
And you're extrapolating from what you do have to work out what you don't have would have looked like.
Yeah, so much of the humerus is preserved, not the entire thing, but I'm a pretty average-sized male in this country. I'm about not quite 6 feet tall, but the humerus of this gigantic penguin would have been about the same length as mine, just to give you a sense of how big this bone is.
You don't weigh 150 kilos, looking at you.
I don't. I weigh almost exactly half that.
But this thing must have been enormous then.
This was an utterly enormous bird. We estimate that its body mass would have been in the range of about 150 kilograms, or I'm from Canada, as you can probably tell from my accent, so back home we'd say 350 pounds. An absolutely huge, huge bird.
Why so big?
Well, there are various ideas about why penguins achieved such an enormous body size range early in their evolutionary history. Some of the most compelling ideas have to do with heat retention. Of course, birds and penguins are warm-blooded animals, and the larger you are, the deeper you can dive, the more oxygen you can hold on to. And these penguin fossils come from southern New Zealand. They would have been diving into very, very cold water, and being very large-bodied would have enabled these gigantic early penguins to have collected more food, to have retained heat more efficiently, and probably also to have dived for longer because they would have been able to retain oxygen longer too.
But penguins still do all of those things today. So why have they shrunk?
Well, penguins achieved very, very large body sizes early in their evolutionary history. So this fossil is about 57 million years old. And enormous penguins persist in the fossil record until around 25 or so million years ago.
Oh, that's significant because the Earth was really hot then. 30 million years ago, the polar ice caps melted, wasn't it? It was really warm then.
That's right.
Was it a case they shrunk because the temperature went up?
I don't think that's the argument I'd go with. There are smaller bodied penguins around at the same time. The important factor here is that around 25 or so million years ago, marine mammals that fall within the same general size range as these very large-bodied penguins come on the scene. And so we think the disappearance of these enormous penguins may have had something to do with the evolution of marine mammals that may have outcompeted these gigantic penguins.
So are you saying they're seal food?
Well, they wouldn't necessarily have been seal food, but they might have been competing for the same food resources. And not only competing for the same food resources, but potentially also the same places at breeding colonies on beaches. Because, of course, animals like seals need to come ashore to have their pups. And penguins, of course, need to come ashore to lay their eggs.
You're saying this is 57 million years back in time. Now, that is a significant point because that's not that long in geological terms after the dinosaurs disappear. So how significant is that timing that suddenly these very large birds are on the scene?
So you're absolutely right, Chris. The extinction of the giant dinosaurs took place, we've got pretty good dates for this now, right around 66.02 million years ago. And the fact that we find these enormous penguins so early in the aftermath of this extinction event, within 10 million years of this extinction event, tells us that birds, once they lose the ability to fly, have the evolutionary capacity to attain enormous body sizes very, very rapidly. So it tells us something fascinating about the maximum possible rate of body size evolution in these animals.
And would it have been the disappearance of the dinosaurs that meant they could get that big because they weren't turning into dinosaur food?
Well, it didn't have to do specifically with the disappearance of the dinosaurs, I don't think. But of course, at the same time, as the dinosaurs, we have giant marine reptiles that would have occupied these macro-predatory marine niches. And I suspect it was the simultaneous disappearance of these giant marine reptiles 66 million years ago that may have opened up available ecological niche space that these birds would have been able to move into.
Is New Zealand special here?
Well, New Zealand is very special when it comes to the fossil record of giant flightless birds, because moa come from New Zealand, as well as these giant penguins. But for penguin evolution specifically, New Zealand is very important because the very oldest penguin fossils we've ever found, as well as a very diverse fossil penguin fauna, have been recovered from New Zealand sediments.
Would this argue then that New Zealand is possibly the origin of penguins?
Well, we think New Zealand and Australia would have separated around 86 million years ago. So New Zealand was an isolated landmass 57 million years ago when this giant fossil Kumimanu was alive. But the evidence that we do have suggests, indeed, that penguins may well have originated in New Zealand.
Amazing thing to end the interview on. Thank you very much. That's absolutely fascinating. Daniel Field there. And if you want to read more about Daniel's research, he's just published that in the Journal of Paleontology. That's it for this week, but do join us on Tuesday next week when we will get a reaction from the UK Science Minister's call for commercial nuclear fusion he says he wants to see by 2040. Could unlimited low-carbon energy really be on the near horizon? The Naked Scientist comes to you from the University of Cambridge's Institute of Continuing Education. It's supported by Rolls-Royce. I'm Chris Smith. He's James Titko. Thanks for listening, and until next time, goodbye.